Hello everyone, and welcome to today's live broadcast, Zebrafish, an upswing model in research and the need for refinement of anesthesia. My name is Valentim from LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots, the leading scientific social networking site and producer of educational virtual events and webinars. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would like now to introduce today's speaker, Dr. Ana Valentim. She is a postdoctoral researcher at the Institute for Research and Innovation in Health in Porto, Portugal. She earned her degree in biology and her PhD in veterinary sciences. Her main line of research is to understand the anesthesia side effects in animal models, namely rodents and now zebrafish. In this sense, she intends to refine anesthesia to be safely used for research purposes. Since 2007, Dr. Ana Valentim is also a trainer at the Laboratory Animal Sciences course, Category C, that's accredited by PhilAsa at IBMC Porto. I will now turn it over to Dr. Valentim for her presentation.
Thanks a lot for that kind presentation. Thank you for having me here. Welcome, everyone. So I'm here to talk about a little bit of the zebrafish, an increasingly used model in research, and the need for refinement of anesthesia. So I want you to say how I'm going to give this presentation. First, I'm going to introduce you to the zebrafish, its uses and advantages in research, how it is seen as a laboratory in the laboratory animal science, why to perform anesthesia in zebrafish and in fish in general, and this has something to do with the evidence that fish really feel pain. I will describe a little bit some anesthetics used in fish, how anesthesia may contribute to the zebrafish welfare, and hopefully you can take home some messages. So, beginning with the animal model, the zebrafish is a small fish, more or less 5 centimeters in maximum. It has a lifespan of 5 years maximum in the laboratory. Of course, that normally the animal caretaker euthanizes that more or less at 3 years. In nature, it survives more or less 2 to 3 years. It lives in shallow waters, but it usually occupies all the water column. It has some pigmentation that may vary with the background, the water quality, and the health status. It also has some sexual dimorphism that you can see here. So here is a female and a male. You can see that the body shape is quite different. The male is more slender, and the female has some enlarged abdomen because of the oocytes inside. The female is not always in this status, of course. And you can also distinguish it by the coloration. The male has some yellowish coloration in the tail fin. But what is important here is the female has a genital papilla that is really useful to identify when you don't see the enlarged abdomen. It's quite easy to see when you put the tank in the light and you can see it. This animal model is quite used in research, and here with a simple search in the Pubmed database, putting zebrafish as a keyword in the abstract or in the title, you can see an increasing in publications in the last 10 years. Moreover, zebrafish has also a journal with its name, so it's really evident that it's a relevant animal model. Zebrafish is used in as an animal model in human diseases. Of course, that it's not a well-established animal model in all these diseases. It's just, we are just refining it. It also used in drug discovery, in gene function identification, for toxicological studies, especially in the ecological perspective, but also for pharmaceutical purposes, and in the regeneration studies. In the regeneration, the tail fin of the fish is highly regenerated, so it's very important. It is also very used in the cardiac research. You can see lots of articles published about this topic, the importance of zebrafish in research. This all happens because it has advantages to using this animal model. So the embryos are transparent, so you can see the development of the fish all over when it's growing. It has a high number of descendancies. A mating pair, you can have 100 to 200 eggs per week. It's a small animal model, so you don't need much space. It has a quick development. So from hatching to free-feeding larva, from fertilization to hatching to free-feeding larva, the zebrafish passes at just 3 to 6 days post-fertilization. And all of this will reduce the cost with the animal. You can see that in this illustrative table. So you can see that in the husbandry infrastructure, the cost per animal per year is decreased with fly and with zebrafish, but zebrafish has advantage more than the fly. There is the genetic similarity and pathological similarity with humans. It also are quite advantage when you want to use large-scale screenings with several drugs. So you have zebrafish and you have rodents, and it's the most used animal model. In terms of laboratory animal science, you can see that in the health screening, the rat and mice is well established. In the zebrafish, there are some guidelines. Hopefully, FUASA will publish some guidelines this year about it. Regarding the environmental enrichment, it's generally recommended for rodents. But the zebrafish is recommended, but it has some contradictory studies and some practical issues, so it's not really well established. For husbandry conditions, for rodents is well established, but for zebrafish there are still huge differences between laboratories. The rodents, for rodents you can find lots of international courses in the laboratory animal science. But for fish, you can see some courses for aquatic animals. But for zebrafish in particular, you just have just a few courses. So I think that we really need to increase the research in laboratory animal science. And also in the topic of anesthesia, where rodents, you have lots of different anesthetic protocols. And for zebrafish, you have some anesthetics for fish in general. But in zebrafish, we have some risks because it's a small animal, and the risk for overdose is huge. Apart from we need to refine anesthesia and the procedures to do better science for the animal welfare, but also we have some legislation that obliges us to do that. So the European Directive already puts the non-human vertebrate animals inside its protection, and also independently feeding larval forms. So the zebrafish, from the time it hatches, it's an animal that is protected by legislation. This was achieved because of the evidences piling up all over the years that pain that fish really suffers pain. There are some evidences that I'm going to describe very briefly. Lynn Snedden has lots of articles about it, and if you are interested in it, please go and see it. So this is a representation of the fish brain versus the human brain. Quite complex, of course, the human brain, but there are some structures that can be analogous. For example, the hippocampus in humans analogous to the lateral pallium. The medial pallium related with the amygdala, and the dorsal pallium related with the neocortex. These three structures in humans have been described as being involved in emotion, in perception, in subjective feelings. So they are very important, and there are some analogs in zebrafish. Moreover, fish has also some opioid receptors in the brain. When they are facing a painful situation, it was detected some electrophysiological activity in forebrain and midbrain. The A delta fibers and C fibers that we found that we can find in mammals, you can find also in the zebrafish and in fish in general. Although our A delta fibers are in less number than the C fibers, and in the fish is the contrary, but it has been discovered that some A delta fibers act as C fibers of mammals. Other evidence is the alteration of behavior of the fish when they face potential painful agents. For example, in this situation, where zebrafish is placed in the tank with two compartments, one is favored because it has some social reward and some plants, and the other one is just a barren environment. So you can see here that it spends more time in the favorite chamber, but when the zebrafish is injected with acid, it started to spend more time in the barren environment because it has an analgesic inside. So it just changes its behavior to alleviate pain. This is a list of criteria for animal pain, and I just want to call your attention for the yes in the fish column, the teleostis fish. So you can see that they fulfill all the criteria of pain. So once more it's proven that we need to do anesthesia, and we need to refine anesthesia for better scientific outcomes. Anesthesia that is adequate for scientific purposes, practical, so researchers will use it, and to protect the animal welfare. This is one example of the stages of anesthesia, and you can see here that the depth of anesthesia is increasing in the scale. There are different scales that you can find in the literature. The maximum of this is 4, some have 5 stages. The 4th stage here is to avoid. It's when the fish is almost dead. It's getting an overdose. So for proper anesthesia, you must do the 2nd stage, and for surgical anesthesia, the 3rd stage has to be achieved. This is just to call your attention to monitor the anesthesia of zebrafish. The anesthesia in fish, ideally, this is the ideal, the best thing it could be. It's fully reversible. You can manage to control the depth of anesthesia, no mortality occur. It must be safe for the animal, but also for you. So the agents used should not provoke risk hazards, be biodegradable, provoke minimal changes in physiology and behavior, and minimal stress, pain, and suffering to the animal. It would be great if it's inexpensive and fully available. In zebrafish, it's especially important that it can be soluble in water because it's a so small fish that injection of anesthesia it's not very practical, so usually zebrafish is anesthetized in a water bath. And should be adequate for experimental purposes. It should depend what do you want to achieve. So you have to consider some variables because the anesthesia, it depends in the duration that you want. So if it's long anesthesia, you have to be careful with the concentration. With the temperature of the water, not only for the fish but also to the onset of action of the drug because it can be different. The pH can also interfere with the action of the drug. To have oxygen in the water. The heart and respiratory rate of the animal can also influence the anesthesia and the way it is metabolized. The body weight of the animal, so smaller animals have more problems with overdose. The gill surface skill is also very important. And of course that the fish has so many species so different from each other that you have to take in consideration that. And also the strain and age of the zebrafish, of the fishes. And once more, it also depends on the experimental purposes. So I really advise to do a pilot study to refine anesthesia, to understand what it works in your situation, what you want to achieve. There are some considerations during anesthesia that you should pay attention before, during, and after. Do not wear powder gloves to handle the animals. Do some feeding restriction 24 hours afterwards, before the anesthesia, or 48 hours if the fish are bigger. Use the water for the anesthetic bath taken from the holding tank, or at least with the same characteristics, the same pH, temperature, salinity. Provide oxygen because it would really improve the recovery, especially in bigger fish. Check also the operculum movements to assess respiratory rates, and check the gill color, a pink light color or a brown color, but pink, it's very important. If the purpose is to anesthetize fish for surgery, test first the response to a painful stimulus. And if respiration, the operculum movement is almost none, so take the animal from the water and put in the recovery tank with clean water, of course. So now I want you to show a movie, and it's a very simple movie, a homemade movie. So you're going to see the fish already in the anesthetic bath, and you have some subtitles so you can follow what is happening. And this is not in the real time. So I'm just going to put it. Okay, I don't think it's working. Okay, I think I'm back. Okay. So this was only to show you how you can monitor very easily the anesthesia in zebrafish. So now I'm going to talk about some examples of some anesthetics that are used in fish. I have to start with the MES222. That's the most common used anesthetic. It's a local anesthetic. It's the only approved by the Food and Drug Administration for use in aquaculture for fish consumption. But it may cause irritation in the mucosa of the fish because it is acidic, so you always need to buffer it, for example, with sodium bicarbonate. It also induces the decrease in the heart rate. It's relatively expensive. It usually is sold in powder, so you have to prepare it first. Except Finkel, that is a brand name for MES222, that is a different formulation that is already in the solution, but you have to buffer it also. So it also increased levels of plasma cortisol and glucose and other physiological alterations. You have also benzocaine, that it's quite similar to MES222. You can find also in different formulations. The hydrochloride is the one that is soluble in water. It has a narrow margin of safety at high temperatures, so it may be more complicated for zebrafish that live at 26 up to 28, 29 degrees Celsius. It induces total immobility at high doses, and it's recommended an exposure of the maximum of 15 minutes. The phenoxyethanol, it's quite popular in the fields and also in the domestic aquariums. It has different formulations also. We have here the phenoxyethanol cell by Sigma, but also the Aquacet. It's more for aquariums, domestic aquariums. It's a bactericide, and it's also used in different other industries for as an insect repellent, to produce soaps, perfumes. It induces light sedation to surgical anesthesia. It's safe to use when the fish are being transported. It's moderately soluble in water. It's inexpensive, but it may increase cortisol levels when the fish is in the water, is in the anesthetic bath, and also may induce some involuntary muscle reflex. The clove oil, as it's taken from the clove, so some people think it's natural, so it may be better. It has as an active compound is the eugenol. In the clove oil, the eugenol may appear in different concentrations, different percentages. But if you buy, for example, in Sigma, you can see which is the concentration of eugenol present in the clove oil, and then you can have some standardization. It also is used in different industries, from perfumes, but also as a local antiseptic and anesthetic. It has a very high margin of safety, but it can have longer recovery periods compared with MES222. Usually you're going to see lots of anesthetics compared with the MES because it's the standard anesthetic for fish. It's inexpensive. You cannot put it in the water directly. You have first to put it in ethanol. It has some risk that methyl eugenol, one of the bioproducts, may be suspected carcinogenic. It has also other formulation. There is a derivative from clove oil. The brand name Acquiesce is isoeugenol. Isoeugenol is similar, it's a molecule similar to eugenol, and it can put directly into water, so it's soluble in the water, but it may induce a slow induction and long recovery compared with anaesthesia. The etomidate and metomidate are hypnotic anesthetics often used in veterinary science. It induces rapid induction and recovery, but there are some risk of muscle twitching. It does not provide analgesia, so it's better when you just want to immobilize the fish, and reduce cortisol levels. This may be an advantage to reduce because cortisol is related with stress. However, it has been discovered that these agents interfere with the biochemical pathways to synthesize cortisol. So it's really messing with the metabolism. So it can be a disadvantage. Etomidate is more effective in alkaline water and at high temperatures. The ketamine, it's not very used in fish in water bath, but in injection. It's a dissociative anesthetic with some analgesic properties. It seems interesting to test it in fish because it has been demonstrated to be hemodynamic stable in mammals. It's more effective at high pH for personal experience, I think 8. But it induces some agitation in the fish after during the induction. And you can also see some behavioral alterations as increasing anxiety, inactivity, some circling, which can be some stressful traits. The lidocaine is also used in mammals. It's a local anesthetic, but it's primordially an analgesic. The formulation hydrochloride is the one that is soluble in the water. It induces a surgical plan of anesthesia in zebrafish at high concentrations, but that may have some risk for the zebrafish. In lower concentrations, it can also induce loss of equilibrium of zebrafish, meaning provoking hypnosis in zebrafish, but it will take longer than anaesthesia to do that. And talking about different components, analgesia, hypnosis, I have to talk about the triangle of anesthesia. It's quite famous. This is the simple way to explain it. Some main components of anesthesia have muscle relaxation, analgesia, and hypnosis. Hypnosis meaning loss of consciousness, then in fish are related with the loss of equilibrium. So, as I said, lidocaine has a high component of analgesia in zebrafish, but it takes longer to induce loss of consciousness. So to help lidocaine to achieve that, we had just an hypnotic called propofol. So this is a model of balanced anesthesia. When we used lower concentrations of two drugs, we put it together and obtaining the desired effects with less side effects. So propofol, it's an hypnotic, as I said. It's often used in mammals, in our anesthetic settings, in surgery settings for humans. It's usually used as an injectable. It's not very much about the propofol in water. It's a short-acting agent, and it has some less cumulative effects, which can be very interesting in a water bath. It prevents peak of cortisol levels, so it has potential to be used in transport, and induce few physiological alterations. However, it's less soluble in water. If we join the propofol with lidocaine, as I said, we have an hypnotic and analgesic producing a surgical anesthesia. Using a balanced anesthesia, it's easy and practical to use. Both are easily available, are practical because you just put it in the water and mix it. You can achieve with this rapid loss of equilibrium and painful response. I'm saying all this about the propofol lidocaine because recently we published an article comparing propofol lidocaine with anaesthetic. So we use, a small concentration of propofol and we put it with high concentrations of lidocaine. There is no differences between these different anesthetic protocols. So we compare it with the standard anesthetic, the anaesthetic. So in what we saw, it was that the propofol lidocaine induced quicker an equilibrium loss and also a quicker induction, of loss of light stimulus and painful stimulus reflex. However, anaesthetic induced a much quicker recovery in these animals. So the only thing that we can say here is that the propofol lidocaine is suitable, so it's appropriate when we want a quick anesthesia and analgesia. For example, for tail fin collection. So you have a bunch of anesthetics, but what to do with all of this? So to wrap up, the anaesthetic is a good anesthetic, especially for short duration procedures, and it obtains a quick recovery. Benzocaine is very similar to anaesthetic, but there are not so many studies in zebrafish. The same happens with phenoxyethanol. The etomidate and metomidate seems to be good, but just for immobility because it has no analgesia associated. The chlorine derivatives, it's also good, but when the long recovery, it's not a problem. The ketamine, it has the disadvantage of inducing anxiety. The lidocaine is classical an analgesic. And propofol is poorly studied in zebrafish and seems to has to be the potential to be used in transport. When we join lidocaine with propofol, if you want to attain a quick surgical anesthesia, you use this combination, which is practical. I wanted to see and to show you what is happening in research in the use of these protocols. So a quick search in the Web of Knowledge database. I put the zebrafish as a topic and the number of anesthetic as a topic. And what I saw is, as expected, the tricaine or anaesthetic is the most used one in zebrafish, then the chlorine and its derivatives, the metomidate and etomidate. Here, the phenoxyethanol has the same number of articles than the etomidate and metomidate, but when I checked the articles, I saw that the word zebrafish was there, but it was not about the zebrafish. The ketamine, the most articles that I found, it was related with the toxicological effects of ketamine in development. The propofol lidocaine article is not here because it was published just this year, and so it's just starting to be more studied. We don't need to attain anesthesia and analgesia, it's enough. So I had to talk a little bit of analgesia. In fish, we have some articles when you study analgesia, just to put the fish in painful situations and see how it reacts. So morphine seems to be effective in rainbow trout because after injecting some acid in the rainbow trout lips, what was happened is that the frequency of rubbing, the opercular movement, and the time to resume feeding was increased. But when morphine was added, these behaviors are all decreased to control levels. For zebrafish, the Analgesia, we have lidocaine that is validated as an effective analgesic for zebrafish. There is not in this table, but morphine and buprenorphine also seems to be effective in zebrafish, especially because the effects of these analgesics were reversed with naloxone. There is an antagonist for opioid receptors. So after talking about the anesthetics, the substances, we need to think, okay, so usually the anesthesia is to improve animal welfare and to decrease suffering of fish. But if it's not properly chosen, the anesthetic may also interfere with the animal welfare. For example, it can irritate fish mucosa, as for example the MES222 if it's not properly buffered. It can also induce rapid opercular movement because of respiratory depression or because it is a trait of a behavior of stress. You can also see piping, twitching, regurgitation, erratic swimming, not in the excitation phase of the anesthetic, because it's one phase as you can see it during the anesthesia, but it can also be related with stress. Jumping when the fish are trying to escape from the water, gill bleeding when the drug caused irritation, or when there are some alterations in the plasma osmolality or an increase in the blood pressure. And also when the fish are facing some anesthetics and it alters its behavior. For example, its preference or its space occupation. Sorry. And about aversion, there's two articles that was recently published that I have to talk about it. So one of them, the zebrafish was placed in a tank with 2 compartments. So you have, the zebrafish have clean water in both compartments as a control, and as expected, the zebrafish spend 50% of the time in both sides. But when hydrochloride acid was added to one of the sides, the red one, you can see here that the fish spent most of the time in the clean water. That is the white, the blue bar. When anesthetics were added to one of the sides, you can see here that isoeugenol, benzocaine, MES222, phenoxyethanol, and lidocaine induce fish to spend more time in the clean water than in the water with the anesthetic. The contrary happened with tribromoethanol and ethonidate, where they behave at control levels. Other paradigma you can see here, also a tank with 2 compartments, where fish have a preferred compartment and it spends lots of time. In that compartment, the analgesia, the anesthetic was added and fish contacted and was conditioned to have the anesthetic in that compartment. Afterwards, the zebrafish was placed in the same apparatus, and what happened is that the trachein, in the trachein, the animals spent less time or almost no time in the preferred preference compartment, while the clove vial in metamidate he passes the same time in both compartments. So these articles, what they meant is that they agree in the version of to MES222. They agree that ectomidate and metamidate may be a refinement. That and one of them has the introduction of 3-bromoethanol as less aversive. You may know it with the brand name Valentim. It has some risk because it can produce some toxic bioproducts. In one of the articles, isoeugenol was aversive, but in the other, clove oil was not. So, of course, there are two different anesthetics, different chemical properties, although similar, but we don't really know if this difference is because of that or because there are different behavioral paradigms. And what is very important is this need to continue this research about the aversion to anesthetics, but also to integrate this data with the clinical effectiveness of the anesthetics. Because if an anesthetic, it's not aversive, but it doesn't fulfill the purpose of anesthesia, of experiment, it does not have any usefulness. So, concluding, I hope you understand the anesthesia importance. It will improve the scientific results, reducing variability, reducing costs, decreasing mortality, and increasing the animal welfare. It needs to be refined. We have MES222 used for everything, and maybe we should have some alternatives. I'm not saying to replace MES222, although some articles said it is aversive, but I'm saying that we need to find more anesthetics and alternatives for different experimental purposes. For example, we have a challenge of long-duration anesthesia, that it's not easy in these animals in the water, in the anesthetic bath, to have a concentration that anesthetizes the animals for long periods without provoking overdose. Usually, when we have to do some intervention with long duration, we have a setup like this with all fishes and also with zebrafish. The fish can stay in the water like you see here, or it can be put in the wet sponge, and a cannula is put in the mouth, and here is delivered the anesthetic solution, and it go out through the gills. And this is similar to some volatile anesthesia. So it is very important to test the anesthetic protocols for the situation that you want, with the age of the water, the temperature, the zebrafish strain, and age. In the end, we can see that MES222, although it seems to be aversive, it can also be a good anesthetic for some purposes. Ectomidate and metamidate seems not to be aversive and can be used when no analgesia is required, and clove violet seems also to be a potentially good anesthetic. But I also have a suggestion to study new protocols of propofol lidocaine and to search inspiration in mammal anesthesia as the balanced anesthesia, because you really need to have different and alternatives for different experimental purposes. So I have to give you here the site for the Zebrafish Information Network. If people are starting to work with Zebrafish or continue to work with Zebrafish, this is very good. It has lots of information and links. So now it is up to you, if you start to using this animal model, to see Zebrafish as a normal animal model that needs research in the laboratory animal science. In the anesthesia topic, that is the topic that brought me here, but also in the husbandry conditions, feeding procedures, reproduction techniques, health monitoring, netting refinement, because we need to transport the fish from one side to other, and usually it's with the net and it's not very comfortable for the animals, and so on and so on. So we really need to improve all this and to see Zebrafish as a normal animal model. So I thank you for watching, and hopefully you have questions.
Okay, well, thank you, Dr. Valentim, for that great presentation. Before we get started on the question and answer session, I would like to remind the audience of how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. So the first question that we have for you, Dr. Valentim, comes from Olapati from Kanan Ministries, and he says, I was at a program where Zebrafish was mentioned as a research tool for hearing loss restoration 2 years ago. So the question is, are lidocaine and propofol combination effective for hearing loss restoration research in Zebrafish?
Okay, so as I've been saying, everything you have to test it in your own experimental model. So you need to achieve analgesia. So yes, it's a good model. You have a quick loss of equilibrium, a quick analgesia, but you have to be in consideration that it takes longer for recovery. So I'm not familiar about the research in hearing loss, so I don't know how long it would take that anesthesia, and it can be or not suitable. But once more, you really have to do a small pilot study and see if it works.
Okay, great. We have another question from Juliana, who asked, what is the best Zebrafish model for diabetes disease?
Okay, so unfortunately, I'm not the person who can answer to your question, because I'm not really into the different Zebrafish models. So I advise you to see the Zebrafish network, and there you have forums, you have lots of other professionals specialized in different areas that you can contact. I'm sorry, so I cannot give you a straightforward answer for that.
Oh, ZSIN is a great resource. Thank you for bringing that up. We have another question. A person asks, how are these refinements adaptable for use in other fishes, Anaesthesia reticulata?
Okay, so I don't know if you think about, if you are talking about the refinements of anesthesia. So usually it could work, it could have nice results, but you have to test it because it has some different particularities each species. So usually it works, but it depends really how much difference it is, this fish compared with the zebrafish.
Okay. The same person also asked, are there any trainings or short courses on zebrafish protocols?
Okay. On zebrafish, so there are some protocols in that website that I showed you. So it's very good also for protocols. Regarding courses, you have a course. I can. So if you can email me, I can send you the information about it. It's a EVM course. It is for zebrafish and medaka in Germany, and it seems to me that it's one of, well, that's the only that I know specific for these animal models. So it seems to be a good resource for that also.
That's great. Thank you for bringing that up. I'm just curious. Do you have any knowledge about the long-term effects of the anesthesia on the fish?
Okay. So I'm still a little bit of a rodent person because I started to work with rodents. So I worked more time with rodents than with zebrafish, and I'm starting this line of research now with zebrafish. And first, I'm starting with refinement, and then it is in my project to check the long-term effects of the anesthetics, namely in the behavior, cognition, anxiety is one of the works. And I also had some works. Our group had some works not published yet about the influence of ketamine in behavior when the ketamine is exposed during development, and there are some particular interesting differences, especially in anxiety.
Okay, great, thank you. We have one more question from Dana, who is from Ontario Ministry of Agriculture and Food, who asks if you can describe piping in zebrafish.
The piping is a little bit like trying to get air from the environment. So just trying to breathe very fast with the mouth. It's more or less that.
Okay, thank you. That's great. Okay, so I think if there are no more questions, I would like to once again thank Dr. Valentim for her presentation. Do you have any final comments for the audience?
Well, I have to state once more that when I changed from the rodent settings to the zebrafish settings, I think it's huge what we have to do and to the research that we have to do in the laboratory animal science in this animal model, because there are not many things, not very systematic things, and I really, it's a little bit disappointing but also exciting trying to research and to understand all of this, and I hope that other people working with zebrafish can also help this research. Thanks so much for having me here. Thanks for watching and for all the questions. And I thank you also the lab roots.
Okay, once again, I'd like to thank you for such a great presentation. So for the audience, today's webcast will be available for on-demand viewing through August of 2016. You receive an email from LabRoots alerting you when this webcast is available for replay. and we invite you to forward that announcement to your colleagues who may have missed today's live event. That's all we have for time for today, and we'll see you again next time. Bye-bye.